After having completed our 4 to 6 hour run on Lake Whatcom, during which all the samples are taken, they are brought back here to the laboratory at the Institute for Freshwater Studies at Western Washington State College and are analyzed further. Here, for example, is the oxygen bottle that you saw being fixed in the field. This was the one that we shook so violently to fix the concentration of oxygen. When the sample is brought back to the lab, it is titrated here against this solution in this column. A known quantity of this sample is placed into a beaker. Poured into this dish. Then into this solution or sample, a quantity of the material in this tube is introduced until the sample has a very light color, as you see here. An indicator is added, which helps in determining the end point of this analysis, and then some additional solution is added until it reaches that point. When this is completely cleared up, we can then calculate from the amount of solution used the exact concentration of oxygen present. This sample contained 6.9 parts per million. Now, the determination of the amount of oxygen present in a given body of water is probably one of the best indications of the condition of that body of water. A low concentration of oxygen is usually indicative of a high concentration of partially decomposed organic material or the superabundance of animals or fish, can be any organism present which may deplete the oxygen. This then would be an unsuitable source of drinking water. A high concentration of oxygen, on the other hand, is usually an indication of a very desirable potable water. The high concentration of oxygen, of course, is also necessary for the support of fish and other desirable aquatic organisms. Now, there were a number of other samples which you saw taken out in the boat. We can go over to the other part of the lab here now and perhaps see what is done with some of these other samples. This area of our laboratory is our bacteriological center. In this region, we analyze some of the samples taken on the lake further. Perhaps you'll recall that in the field, some of the samples were simply collected and no particular manipulation was involved in the field. Have here one of these samples. The only particular thing about these samples is that they were taken under very sterile conditions. Nothing was done to contaminate them. Once back in the lab, they are filtered. This small disc is a very fine pored plastic filter. Place this here. Place a funnel on top, so, and add a known quantity of this particular sample. Then the water is drawn through the filter using a vacuum pump, as you can hear in the background. When the sample has been drawn through the filter is removed and placed in this small dish, like so. It's covered and then incubated for 18 to 20 hours. Now, after the filter has been incubated, we are able to count the number of colonies of bacteria which are present. Here, for example, is a filter which had been incubated for 20 hours, and perhaps you can see the small dark spots on this filter. Each of these spots represents a colony of bacteria now, and we can say that for each colony present on this filter, there was one bacterium present in our sample. This then gives us a very accurate count of the number of bacteria present in a known sample. And with this number, we can plot the distribution of bacteria in Lake Whatcom and watch the seasonal changes and changes which occur from station to station in bacteria concentrations. Now, we have some charts here which will better serve to illustrate some of our findings on the concentration of bacteria. Here we have a map of Lake Whatcom, showing here from Silver Beach, Geneva, Strawberry Point, here on down to the south end of the lake. And here on a bar graph illustrated the concentration of coliform organisms present in these various regions. Now, before going further, perhaps we should decide exactly what coliform organisms are. They are a group of bacteria, most of which are characteristic inhabitants of the lower digestive tract of man and certain other animals. Now, in themselves, they are not particularly harmful, that is, in small quantities. They are, however, indications of, or indicators of human fecal pollution. Therefore, if we find coliform organisms present, we know that, potentially at least, such organisms as those which cause typhoid fever, or cholera, polio perhaps, infectious hepatitis. These also could be present, so a measure of the concentration of coliform organisms is very important. Now, we can see from this map and the accompanying graph that the concentration of coliform organisms is highest here in Impoundment 1, that is around Geneva, Lodell Donovan Park, and in Impoundment 2, that is from Geneva to Strawberry Point, and in the region where the city water supply is taken out. As you proceed southward down the lake, the concentration drops off very sharply. Here, just across the sill from Strawberry Point, it drops off very sharply and is at a very low level throughout the remainder of the lake, except that it is somewhat high here in the region where the inlet waters, that is, the waters from the Nooksack River, enter Lake Whatcom. Now, the high concentration here, these two bars, is explainable, certainly, on the basis of the large number of people that live around this part of the lake. This is just the sort of thing that you would expect. The high concentration here at the inlet we have traced to pollution by cattle, largely. So this is perhaps somewhat misleading.